Effect of nicotine and atropine on the frog's heart. Principle. Nicotine and atropine are neurotransmitters which affect the frog heart rate. We set up the experiment similar to the previous experiment. We stimulate the vagosympathetic trunk of the frog and record the resultant change in the cardiogram. Now we stimulate the white concentric line which is present between the atria and the ventricle of the frog. We record the resultant graph. Now we stop the stimulation and allow the heart to return to the normal cardiac rhythm. Then we apply nicotine solution to the heart. Again we stimulate the vagosympathetic trunk and observe the resultant graph. Then we stimulate the white concentric line and observe any change in the graph. Now we wash the heart with normal saline to remove the nicotine. Now we apply atropine to the heart. We stimulate first the vasovagal trunk and record the resultant changes in the graph. And then the white concentric line and note the changes in the graph. Finally, we study all three graphs and analyze the findings. In the first graph, when no drug is applied to the heart, when we strongly stimulate the vagosympathetic trunk, the heart stops beating for some time. When we stimulate the white concentric line, the heart stops beating again. Now let's look at the graph after the addition of nicotine. In this graph, we can see that stimulating the vagosympathetic trunk has no effect on the heart. The heart rate and force of contraction remain normal. But when we stimulate the white concentric line, the heart stops. In the third graph, which shows us the effect of atropine on the heart, we can see that stimulating the vagosympathetic trunk or stimulating the white concentric line does not affect the heart. The heart keeps beating at the normal rate and force. Let us understand why this happens. Let us first understand the parasympathetic nervous system. The parasympathetic neurons that originate from the spinal cord are long neurons. These neurons travel through the body to the target organ and synapse at a ganglion near the target organ. We call these neurons preganglionic neurons. From the ganglions near the target organs emerge short neurons which directly synapse onto the target organs. These are called postganglionic neurons. The neurotransmitter in the parasympathetic system is acetylcholine. The preganglionic neurons secrete acetylcholine at the ganglions. This acetylcholine acts on the nicotinic receptors on the postganglionic neurons and stimulates the postganglionic parasympathetic neurons. These neurons in turn secrete acetylcholine at their nerve endings. The acetylcholine acts on the muscarinic receptors on the heart muscle cells and through inhibitory G proteins associated with the receptors reduces the force of contraction and the heart rate and stops the heart from beating. In the first graph, when we stimulate the vagosympathetic trunk, we are stimulating the preganglionic parasympathetic nerves which stimulate the postganglionic parasympathetic nerves which inhibit the cardiac muscle cells. When we stimulate the white concentric line, we stimulate the postganglionic neurons directly. Now let's see what happens to the neurons in the presence of atropine. Atropine blocks the muscarinic receptors of the heart muscle cells. Thus, stimulating either the preganglionic or postganglionic cells produces no effect on the heart muscles. Thus, the cardiogram remains unaffected by stimulation of the vagosympathetic trunk or the white concentric line. Now let's see what happens to the neurons in the presence of nicotine. Nicotine inhibits or blocks the nicotinic receptors on the postganglionic neurons. So when we apply nicotine to the heart and then stimulate the vagosympathetic trunk, the impulse travels through the preganglionic neurons but is not transmitted to the postganglionic neurons. The nicotine blocks the nicotinic receptors. When we stimulate the white concentric line directly, we stimulate the postganglionic neurons directly. This impulse reaches the heart muscles and thus the heart stops beating. We can see that stimulating the vagosympathetic trunk has no effect on the heart. The heart rate and force of contraction remain normal. But when we stimulate the white concentric line, the heart stops. Through this experiment, we can prove that the site of action of atropine is the muscarinic receptors on the cardiac muscle cells, and the site of action of nicotine is the nicotinic receptors in the ganglion.